Hi, I'm Matt, lead developer on IDPicker. In this tutorial/glossary, I will introduce vocabulary used in the IDPicker 3 application and in other tutorials. I will assume you're already familiar with proteins, peptides, modifications, mass spectra, and charge states. A spectrum source, sometimes just called a source or run, is the raw data, the mass spectra, from a shotgun proteomics run. It can come in many formats that IDPicker supports, including vendor formats. Spectrum identifiers are unique within a source, but not between sources. A spectrum source group, sometimes just called a source group or even group, is a user's arbitrary assignment of spectrum sources to an experimental hierarchy. The example here groups a couple technical replicates into groups named according to cell line, but the hierarchies can be arbitrarily complex. A distinct analysis is what you get when you identify your data with a given software with a specific name, version, and parameter set. Usually, you run the same software with the same parameters on many different spectrum sources, but it's not unusual to combine different analyses in a single IDPicker report. This can be due to multiple runs with different software, or the same software run with different parameters. IDPicker is able to group the identifications from each run according to its analysis. One example here is showing how a small difference in the mass set in dynamic mods can result in a distinct analysis. A distinct peptide is simply a distinct sequence of amino acids. In particular, modifications and charge state are disregarded. However, different isobaric sequences do count as distinct. A distinct match is a distinct peptide plus the charge state and the modification positions and delta masses. Just like with distinct analyses, it can be important for the dynamic mod masses to be configured equally so that small mass differences are not counted as multiple distinct matches. My favorite part, protein assembly. IDPicker has an easily comprehensible approach to this difficult task. The different results of the assembly process are color-coded here in this diagram, which is technically called a bipartite graph. The proteins in the red boxes indicated by their accessions are grouped together as a protein group when they are evidenced by exactly the same set of peptides. So all of these proteins in the first group map to the first 4 peptides. The second group has an extra peptide, a unique peptide, which makes it a separate group. But it doesn't have 2 of the first group's peptides, so the first group is not a subset of the second group. The blue boxes are the peptides, and they are grouped together as a peptide group when they are evidence of exactly the same set of proteins. The green boxes are clusters, a way to organize the proteins and peptides into associated subsets. Since there are no connections between the first 2 protein groups and the last 3 peptide groups, 2 clusters are created. A peptide spectrum match, PSM, is the basic unit of evidence processed by IDPicker. It is the unique triplet of peptide, spectrum, and analysis. Peptide spectrum analysis match was just too wordy. PSMs are given a rank depending on their scores, and what you usually see in IDPicker is just the top-ranked PSMs and their associated proteins and peptides. In the example here, the combination of multiple analyses and isobaric peptides results in 8 different top-ranked PSMs for just 3 spectra. You can see that X-tandem and mirror match each failed to identify one of the spectra, otherwise it would have been 12 top-ranked PSMs. A filtered spectrum is one that has passed IDPicker's filters, which are covered in another tutorial. Those filters aren't just at the PSM level, it includes the effects of protein level and peptide level filtering as well. As you can see in the example given here, isobaric residues can often lead to ambiguity of the identifications of a spectrum. All 3 spectra provide equal evidence for both peptides, and thus also to the 2 protein groups highlighted in red. Data filters in IDPicker are important because it allows you to filter the report to get the subset of results you want to look at. There are 2 kinds of filters. The basic filter is applied to the IDPDB to create an internal subset of top-ranked PSMs and their associated proteins, peptides, and spectra. The features of the basic filter are covered in another tutorial. The view filter doesn't modify the IDPDB, it just changes the way the database is queried and how the results are presented. You can create a view filter by double-clicking most rows in the various views, or by selecting multiple rows by Control or Shift-clicking and then pressing Enter. View filtering is covered in detail in another tutorial as well. Layouts in IDPicker allow you to present the report in a format that works best for your goals. You can drag and drop the views to dock or stack them, or you can press the X button to hide them as a tab at the bottom. There are 2 places that layouts are stored, in the IDPDB itself and in your user settings. The layout menu lets you save layouts to and load them from both places. For example, if you want to send your IDPDB to someone and have them see it a certain way, save your layout as the database default. If you want to always create new reports a certain way, save your layout as the user default. If you just want to have some alternative non-default layouts easily accessible, save them as named layouts either in your user settings or in the database. Layouts are covered in detail in another tutorial. Tree grouping allows you to change the structure of a view from a flat table to a hierarchical tree view. Because the flat form of the protein view is an individual protein and the basic row of the peptide view is a distinct match, tree grouping is necessary when you want to get one row per protein group or one row per distinct peptide. The basic row of the spectrum view is a PSM, so tree grouping of some kind is usually necessary until the data is narrowed down a great deal by a view filter. The values of some columns change automatically based on the tree grouping. For example, if you group the spectrum view by spectrum and not by source group and source, then the spectrum ID will include the source group and source name so that the spectrum is still uniquely identified. Pivot columns are extra columns you can turn on for some of the views that add more targeted summary information, such as showing spectral counts per spectrum source in a protein row, or distinct matches per spectrum source group in a peptide row. These extra columns are used to provide approximately the same tables in IDPicker 3 that IDPicker 2 provided by default. That's it. Now you know your basic vocabulary for IDPicker, and knowing is half the battle.